Okay, so we are recording. Thank you very much. Great. So we are here today at the first of a series of journal clubs that we are going to have during the summer. We're going to have three young investigators from different areas in the US, but all of them working on retina, on different aspects of retinal physiology in health and disease. The first one of our guests is Dr. Levi Todd from Upstate Medical University in Syracuse. Levi is an assistant professor in ophthalmology and visual sciences. He did his PhD at the Ohio State University in 2017, and then he completed postdoctoral work with Tom Ray in University of Washington in 2023, and he's a very new assistant professor at Syracuse. He's very close to us physically, but also scientifically, he's working on things that are really, really cool and how to regenerate the diseased retina using glial cells. So I will shut up and please share your screen and let's dive in.
Yeah, thanks. Let me do the share. We know it worked. We tested it. Oh, hold on. I lost my PowerPoint.
Yeah.
Okay, I'm good, right?
Yeah, yes, good. I can see.
Okay. Yeah, thanks, Michael. I'm excited to do this. Yeah, so as Michael mentioned, I am trying to do what doesn't happen normally in the retina, and it's after neurons are lost, they're gone forever. And what we're trying to study is ways to maybe develop cell replacement therapies to replace neurons that are lost that can lead to blinding diseases, or that cause blinding diseases. I like to give this PSA every time to remind people that, the retina is the brain. It's not some peripheral nervous system structure, but during development, the midbrain literally bulges out from the neural tube outside of your skull, and you have pieces of your brain floating outside of your skull in the back of your eye. So this is brain tissue, the retina is. And because it's brain tissue, it means once neurons are gone, they don't come back. So this nice, beautiful picture from Rachel Wong's lab shows the different layers of the retina. This group knows this is a beautifully organized neural structure. When any of these classes of neurons die, they lead to blindness. And, when ganglion cells die, they lead to things like glaucoma. When photoreceptors die, things like macular degeneration. So as I mentioned, the retina is CNS. And we've known for a long time this depressing fact that, depressed Gahal, as you can see in this picture here, which is, once the nerve paths are fixed, when they die, nothing's regenerated. And, Gahal had this beautiful quote that it's like for the challenge for future scientists to change this harsh decree. So I think, how we see our work is, 110 years later, trying to kind of take up this challenge of fixing the broken mammalian brain. So the reason we think this isn't science fiction is because this already happens in nature. Other vertebrate species can regenerate their retinas. And their retinas look, very similar to human retinas and very similar to mouse retinas. This cartoon here on the left is next to a section of a zebrafish retina labeled for HUCD in blue here. So HUCD labels the ganglion cells of the zebrafish and the interneurons, the amacrine cells. This time course you're seeing here is days post-injury. So they can inject a toxin into the zebrafish eye, kill all these neurons. And you can see in the period of a week to 2 weeks, these neurons come back. This is functional regeneration. These fish can see again. People have done behavioral testing. They've done ERG and light response assays to show the zebrafish have functional regeneration. And it was known that fish could do this for 50 years, but it wasn't known the source of regeneration until we recently developed transgenic lineage tracing. Well, not recently now, but 20 years ago. So Dan Goldman's lab had shown in University of Michigan, they had shown that the Mueller glia are the source of regenerated neurons in fish. So when you develop a transgenic lineage tracer, you can label the Mueller glia here, this big green cell here, and you can see that the neurons that are regenerated come from the Mueller glia. So all vertebrate retinas have Mueller glia. Humans have Mueller glia, fish, just like fish have Mueller glia. And Cahal loved to draw these cells, these beautiful cells that span the entire layers of all the retina, and they do basically everything you've heard astrocytes do in the brain. They can recycle neurotransmitters, they can couple vasculature with activity, and they can do things to protect the retina and the brain and maybe hurt the retina and the brain if they become too inflamed. In the retina, they also do some very specific things, like they can help recycle the opsin from the cones. And some investigators think they might actually serve as light guides to the photoreceptors. But these cells are really amazing. These are the Swiss Army knives of the retina. I think these are very beautiful cells to work on. But what we're interested in, at least for this talk, is they can be a source for regeneration in some species. So in fish and frogs and probably salamanders, people are finding now, these are the cell types that are used for a source for regeneration of neurons. So this is how it works in fish. When the zebrafish sense retinal injury or degeneration, these Mueller glia reprogram, they re-enter the cell cycle, they give rise to these what's called transit amplifying progenitor cells. And these cells go on and give rise to all the neuronal classes of the retina. So they regenerate all the major types of neurons in the retina, and these fish can see again. Now, we know this because just like humans, mice don't do this at all. There's no spontaneous neurogenesis. When mice Mueller glia sense injury, or in humans, they respond with inflammation. People call this gliosis, but it's a complex response, and it can be releasing inflammatory cytokines. They become tense and stiff, and they can upregulate fibrotic molecules. But no neurons are born in the adult mammal. So, what we're interested in as a field and in my lab is, why doesn't this happen in mammals? What happened maybe in evolutionary space and time to lose regenerative capacity of glia? And could we force this to happen? And one of the ways we think about this is, can we just understand potentially how this happens in other species? And a lot of groups do this. There's a lot of really good labs across the country that are studying how zebrafish Mueller glia can regenerate neurons. And this is a huge field, and there's everything you probably know about retinal development is reactivated in regeneration. So all of these signaling pathways come up, the wince, the notches, the hedgehogs, all the things we know about building a retina need to be reinitiated to rebuild a retina. So just to skip maybe 10 years of work here, what I'm going to focus on here is, we had focused on this gene called ASCL1. So all of these signaling pathways kind of converge on a few proneural factors which are required for regeneration. This is a transcription factor that is required for zebrafish to regenerate neurons. So all of these signaling pathways eventually need ASCL1 to be present. If you knock down ASCL1 in the fish, you don't get regeneration. Okay, so what we had thought in the past is, could we just kind of force these regeneration genes in the mouse glia? Because the mouse Mueller glia do not upregulate these genes after injury. They upregulate the inflammatory responses. They don't turn on genes like ASCL1. So we started doing this in vitro at first, and what we would do is we would make primary culture of adult Mueller glia and overexpress with lentiviruses different transcription factors for neurogenesis. And again, I'm skipping a lot of screening. We screened a lot of different factors we thought might matter, but we found ASCL1 could stimulate neurogenesis. So what you're seeing here is a picture in vitro of a primary Mueller glia from a P12 mouse retina. So after neurogenesis is over, we'll culture the Mueller glia. This is with a transgenic lineage tracer, so we know they came from glia because they're expressing this RFP, which is hooked to a glia Cree. So we have glia-derived cells. When we apply ASCL1, we can see they incorporate EDU, which means they re-enter the cell cycle. It's a proliferation marker. And now they express neuronal genes like MAP2. So if you just hit glia with GFP, these quantifications here, none of them turn into neurons. So TUJ1 is a neuronal marker, MAP2 is a neuronal marker. But if you hit them with ASCL1, about 30% of these glia will turn into neurons. This is really exciting, but it was just in vitro. And it was just young mice. That's when we started making these really complicated transgenic mice that I'll go through because they feature a lot in this work. We wanted to be able to activate ASCL1 only in glia in adult mice. The way we do that is through this triple transgenic mouse. And to keep it simple here, what we do here is we have a Cree recombinase hooked to a glia promoter. So this is glassed. When we give the mice tamoxifen, it goes and cuts out the stop and turns on a tetracycline activator, which then can activate a TETO ASCL1 in GFP. So to keep that simple, when we add tamoxifen to mice, it turns the glia green and activates ASCL1. So we can lineage trace glia and turn on ASCL1. What we have found is if we just turn on ASCL1, it doesn't cause regeneration at all. But if we do ASCL1 with a retinal injury, you can get these glia-derived neurons. So here's some pictures here of glia, these long cells that span the whole retina, and then where the arrow is pointing, you can see these neuronal morphology cells that kind of bud off of the glia. And this is showing you a neuronal marker of a cell. So here's a glia-derived neuron expressing OTX2 in the middle of the retina, which is a bipolar neuron. This was really exciting for us. Again, this is in vivo regeneration in the mouse retina, but it only worked up until P12. It didn't work in adult mice. This sent us through another side story I'm not going to tell today about chromatin, and we were studying how chromatin changes in development. And what we eventually ended up finding is that if we just do epigenetic modifications, we can cause regeneration in adult mice. So what we were doing then is turning on ASCL1 in glia, and now we were damaging retinas and applying HDAC inhibitors. So HDAC inhibitors open chromatin, and it was allowing us to regenerate neurons even in adulthood, mice that were 3, 4, 5 months old. So here's how this looks. With no injury in this C panel here, you have just the Mueller glia expressing their green GFP. If you do a damage model, this is an NMDA damage, so it kills the retinal ganglion cells. If you do an NMDA damage model and give them an HDAC inhibitor, you can now see these neurons that butt off the glia, these big circle cells. And you can see in these zoom-ins here, you can have cells that don't express the glia marker and now express a bipolar marker. And you can see a couple different morphologies of these regenerated neurons. Okay, this is a really big claim because what we're claiming here is that we're getting in vivo regeneration of neurons in the adult nervous system. So other groups were also finding this in similar ways, and they had to retract papers because of technical artifacts, because they were accidentally labeling normal neurons and thinking they were from glia. So I want to show you just like 3 slides to show you how we are confident this is like bona fide real regeneration. Okay, the biggest things for us is that we are lineage tracing these cells with a genetic lineage tracer. Not only that, we can apply EDU in vivo and show they're newly born. So EDU labels newly synthesized DNA. Neurons are always plasmatotic. They don't go through the cell cycle. So if you just give EDU to an adult mice, you're not going to see any EDU positive neurons ever. What you see in this picture here is a GFP lineage trace glia, and you can kind of see next to it these red and green cells. These are EDU positive cells budding off at the glia, now expressing OTX2 and neuronal marker. So we can catch these cells that went through the cell cycle and are lineage traced. So we're confident they're from glia and they're newly born in the adult. We've done a lot of single cell sequencing across different time points of regeneration. Why that's really important is because we can track cells in transition molecularly. So this is showing down here a wild type mouse after injury. These blue are the Mueller glia. When you see these days post regeneration treatment, you can now see this red cluster, which is a bunch of progenitor genes. And then finally this yellow cluster, which is the neurons, the OTX2 neurons. So if you look at these plots over here, you can see GluL. All these are glia cells. They go through this neurogenic. This is a neurogenin transcription factor for neurogenesis. Then they turn into these OTX2 neurons. So we can catch cells in between, glia, progenitor, progenitor, neuron, molecularly in between. So we know this is really happening from glia. And then lastly, we've had collaborations with Fred Ricci to show these cells can actually make synapses and they integrate into circuitry. So when you shine light steps on a patch clamp Mueller glia, you can see the response of depolarization here. If you shine light on a bipolar cell, you can see how they respond. When you shine light onto a Mueller glia-derived neurons, you can see they respond like a neuron. They respond to light, but it's not as good. So what you can see here is like a 10 millivolt depolarization or hyperpolarization of these bipolar neurons. And you can see the glia-derived neurons are 1/5 of that response. They're these hybrid in-between glia neuron-like cells. We've done a lot of evidence I'll show you later for this. This was exciting because it showed these new neurons actually make synaptic connections because these are not responding to light themselves. They're responding to light because they make connections with photoreceptors. Okay, so this really opened up this whole new idea. Like, can we use glia for a cell replacement strategy of neurons in the adult retina? And there's a ton of questions that like I went to ask in my lab and our field needs to ask in general about how do these new neurons wire into circuit? How do we control the types of neurons that come in? Will they ever actually be useful for vision? And we want to know, can they be used in disease contexts and things that would really matter for this idea of cell replacement therapy? Okay, so I work on a lot of these topics. I work on a lot of neuroimmunology stuff, and I work on a lot of cell fate choice stuff. I'm going to tell a story today about one of the things we've worked on to get different types of neurons from glia. So when we first showed this example of glia, Mueller glia being able to make neurons, we got one kind of neuron, the bipolar neuron. So, you know, clinicians were like, oh, this is really exciting, but it's kind of the most boring neuron you could have got. Like really people want photoreceptors and they want ganglion cells. There's not this like huge, you know, swath of blinding diseases from bipolars. So we were like, okay, this is cool. We're getting in vivo neurons, but how do we control the cell fate that comes from these things? Can we get ganglion cells from these cells, and can we get photoreceptors? That's really the clinically relevant subtypes that probably people are wanting us to try to work on. So, this makes us think like developmental biologists and understand, well, how do you control cell fate choices? So I think this group mostly knows, but in development, how this works is there's a progenitor cell, it's multipotent, and it can make ganglion cells, and then it can make, cones, and then it makes, the amacrine cells, and then eventually it makes rods and bipolars and Mueller glia. So what happens though is there's this like alphabet soup of transcription factors that get turned on at different times of development to basically give the cells the code on what kind of neuron to make at what kind of time. So early on in development, you make ganglion cells. There's particular factors that specify ganglion cells. And later on in time, you make rods. There's different factors for rods. If you look closely, this gene ASCL1, it's up here. I don't know if it's my pointer, but it's one of these late progenitor factors. So it kind of made sense, if you think about this as a clock, we're kind of making Mueller glia go back one step into basically bipolars, which is one of the last things they were already making. So maybe it was a little bit easier to change the chromatin just to go one step back instead of all the steps back to a ganglion cell. So what I wanted to think about was, how do I get the factors to make a ganglion cell into these Mueller glia, and what factors really should we use? So this had us think about, how are ganglion cells made in the mouse? There's a huge literature for this, but now there's a lot of new, molecular biology techniques that can give us a really good resolution of how this works. So what I wanted to ask was, how are retinal ganglion cells made normally? So we take embryonic day 14 mouse retinas, which is about the peak of when ganglion cells are being generated. I sort out all the cells, and we do single cell ATAC sequencing. So what that does is shows you the open chromatin of cells as they're making cell fate choices. So this is what it looks like. This is a UMAP plot, but instead of RNA, this is ATAC sequencing, so it's chromatin. So cells are clustering by each other because of their open chromatin profile. And what you find here is what you'd expect. You have progenitor cells, they go into these neurogenic phase, and they can either make ganglion cells, or at this stage of development, they make cones and horizontals. What's cool about this technology is that you can basically tell the software, here's the path. You make a progenitor cell that goes in to make a ganglion cell. It's called pseudotime. Basically, it's fake time. We're telling in this one time point, we're saying, here's the start, here's the end. We want to ask the software is, tell me the open chromatin down that path. That's what this looks like. So in this plot here, what you're looking at is how the progenitor cells, the chromatin landscape they go through to make a retinal ganglion cell. That's really important because chromatin is open usually when a factor is active. So basically, if you see SOX2 is open here, it usually means SOX2 is pioneering motif sites because it's being functionally active. That's the assumption. So, but what you can see here then is there's a very distinct waves of different phases of factors between progenitor cells to neurogenic precursors to retinal ganglion cells. And you look at this long enough, and you start to see kind of like codes. I don't know if these are real or not, but we have these classes of factors seem to be activated at certain times. So in the progenitor cells, there's a lot of these homeobox domains like RAX, SOX2, VSX2, things that are very famous. When you go into a neurogenic precursor, usually turn on BHLHs. That's like ASCL1 and neurogenin. And finally, when you're a fate specifying factor, these are usually like limb or POW domains. In this context, you have stuff like POW4F2, which is the brain 3B, islet1, or EBFs. So we'd like to think of these as potentially kind of like the menu of what we could maybe use to engineer regeneration. Because we know ASCL1 worked really well to become a neurogenic precursor. We have a different story using some of these other neurogenic factors. We know they work. So now we want to ask, can we use these fate specifying factors to kind of engineer where the cells land in regeneration space? So luckily, MuLab had already shown how 4F2 and islet1 are critical for development of retinal ganglion cells. And really nice, he already had a mouse he generated that was a TET-O POW4F2 islet1 activator. So I could cross those with my mice and make this like Frankenstein quadruple transgenic mouse. Then now basically I can activate ASCL1, make the glia green, and then now turn on these ganglion cell factors, POW4 and islet. So we call this the IPA treatment because that's what everybody drinks in Seattle, but also it helps with the acronym here. So I might call this the IPA cocktail, but that's islet, POW4F2, and ASCL1. I'll try not to be jargony. Okay, so what we're going to do is ask our regeneration experiments, but now activating those ganglion cell factors in the glia. This is just showing that our mouse works, but you can see these green glia that are like changing their morphology during regeneration. And now you can see glia expressing a ganglion cell factor, brain 3, they shouldn't normally express, but it's transgenically expressed. You can see glia expressing islet 1 and sometimes both. The way I should mention this mouse was kind of annoying because it was made in a brain bow cassette. So we activate usually brain 3 or islet 1 or both. We can't really control that. So we're kind of doing 3 experiments in one here with these different factors. Okay, but now we wanted to ask in regeneration what happens when we do this. First, it was really exciting because when you look at these cells from glia, they don't look like glia anymore. They look like neurons. And now they're kind of in different layers and different morphologies. So this is lineage tracing regenerated neurons. And now we are finding they're not just bipolars, which is exciting for us. So now you can see these lineage trace new neurons that are expressing HUCD. HUCD labels the amacrine cells and the ganglion cells. But it's not bipolar. So that was the exciting thing here. So you can see here in the quantifications down here, in gray is ASCL1 only, and you just get bipolars with that context. And you get a little bit of amacrine cells. I didn't mention this earlier, but that's the other HUCD cells. With IPA treatment, this islet, PAL4, and ASCL1, you still get the bipolars, but now you're getting this new kind of neuron, this HUCD cell that we're going to call ganglion. I'm going to try to defend here in the next couple of slides. But a new type of neuron came from a new cocktail of factors is the key. Not only that, they have different morphologies now. They look kind of like ganglion-like morphologies. So when you take these retinas down on a whole mount, you can see these glia-derived neurons in green. They express brain 3, which is kind of cheating because that's transgenic. But now you can start to see these complex neurites come from these cells. This is something we don't see with ASCL1 only. So not only are they looking like they express different markers at the IHC level, but morphologically they look different. So we wanted to do single cell RNA sequencing. So what I do here is I sort out all those green cells I showed you during regeneration time points. So this is what this looks like here. You have all the mular glia here in red. So that's expressing a glia gene like RLBP1. They go through the bipolar phase like I already showed you, and that's this little arm here. Now down here in the bottom left, you can see ASC1 only just gives you glia to bipolars. But when you do this new treatment, IPA treatment, you can now see it makes a whole new cluster of a neuron type, this green cluster down here. So you can see at the transcriptomic level, we're getting a new type of neuron born from these glia, and you can ask what cell or what genes are expressed by those cells, and there's ganglion cell genes there now. There's genes like SOX11, SOX4, EBFs are now expressed in this new cluster here, suggesting these are similar to retinal ganglion cells at the transcriptomic level. We'd also see at the protein level some retinal ganglion-like factors expressed in these glia-derived neurons. So here is a stain for SATB1, which Josh Shane's lab had shown as an off-on direction-sensitive ganglion cell. But you can see here this big green cell from a glia now expressing SATB1, a ganglion cell-specific marker. But look, it's in the wrong place. It's literally where the photoreceptors live. There's another reason that we know this is real regenerated neurons, because you wouldn't get a ganglion cell out where the photoreceptors are. So we're making cells that are kind of confused. They're expressing ganglion cell factors, but they don't really know their location, but they're at least expressing specific things that look like RGCs. We want to see how similar are these cells to real ganglion cells. So what we would do is take a single cell sequencing data set of the embryonic retina. So this is an embryonic day 14 retina for single cell RNA-seq done by Brian Clark when he was in Seth Blackshaw's lab. And you can see the progenitor cells that go on to make the types of neurons at E14. What we do on the right side here is we take the transcriptomes of our regenerated neurons and then try to cluster those with the embryonic neurons to ask, what are you most like transcriptomically? And what you can see here is these regenerated neurons are these green cells superimposed. So these green ganglion cells from the mular glia cluster with embryonic retinal ganglion cells. It gives us a little bit of evidence that they're similar transcriptomically. They share enough genes that they'll cluster in space together as the same cell type. What's also important to see is they don't cluster with the mature ganglion cells. This like end of the tip here, they really cluster as immature state. And that's going to be a big point to make in the next few minutes. But at least gives us confidence that not only they look like ganglion cells by the morphology, they express them in the proteins, but if you look at all the transcriptome, they're most similar to embryonic retinal ganglion cells. So because we started this story with the chromatin, we wanted to look at how does chromatin change during regeneration? And how's that different, or how's that similar or different than development? So what we do is we do our regeneration experiments to make Mueller glia into the retinal ganglion cells, and then ask, what does that look like at the single cell ATAC sequencing level, at the chromatin level? And just like the RNA, you can see you have a big cluster of Mueller glia. Some give rise to the retinal ganglion cells. The others give rise to the bipolars. And you can see these motifs like brain3B is active in the cells that look like they make retinal ganglion cells, suggesting that this factor is one of the main ones driving the ganglion cell fate. But here's what I think is really cool and kind of keeps me up at night still, is how is development like regeneration? People have asked this question for centuries. And this is kind of a cool technology now to look maybe at a very, fine resolution to compare regenerative processes to developmental processes. So on the left side here, this is development. I already showed you this plot. This is asking how the progenitor cells make ganglion cells in normal development, asking all the chromatin opens along that path. On the right here is asking, what does it look like when the Mueller glia make a ganglion cell? What does that chromatin look like on that path? And you can see it's quite different. You like squint your eyes and you like are really hopeful. There's some similarities here. Like there seems like there's a little bit of a path that looks like a retinal ganglion cell, but it's really different. And the biggest thing that's different is that we don't shut off all the glia progenitor genes. So if you look like here, you can see it goes from red to blue, showing all that progenitor chromatin shuts down when you make a real ganglion cell. When you do this in a reprogramming state, they don't lose the memory of being a glia. They keep the glia chromatin open. And they don't eventually activate all of the downstream factors as strong as they should. And maybe that's because all the progenitor chromatin is still kind of hanging around the memory of these cells. We're calling this like glia hangover. And it's not only the chromatin, but even at protein level. So SOX2 in the retina is usually in Mueller glia. It's also in cholinergic Hammerkin cells, but it's not in ganglion cells. So what you can see here, though, is some of these big circular neurons that we regenerate that look like ganglion cells in a lot of ways still express SOX2, a glia factor, which is, again, evidence this is real. This is really from glia, but it's imperfect. It has this memory of being a glial cell. Now, despite this, they are looking exciting. Like we have collaborated with Frederici again, and he would patch clamp these retinal ganglion-like cells from us, and they fired action potentials in response to light, which is really exciting for us because one, in the retina, that's what the main cell type that does as are ganglion cells. And it means, again, they're making synaptic connections to the circuitry to respond to activity. These were not iPRGCs. We knew that from at least the single cell transcriptomes. It seems like they can make, light-informed activity outputs. And we were getting both on and off responses from these cells. But again, a lot of these were immature. So a lot of these cells don't fire action potentials. A lot of them would have these little tiny like micro spikes, and most of them are not going to respond to light. So I don't have the plot here, but it's still a very small subset of these cells will actually do this. So it's still quite incomplete. Nevertheless, I kind of think this story gives us a little bit of hope, that we're not locked into one kind of neuron. We can use lessons from development to kind of figure out how maybe to engineer these glia to make the cell types that we want. So I think in principle, we still have a lot of work to go to make a better retinal ganglion cell for sure. But I think it shows that if we take different factors, we can get different cell fates from these glia. And the biggest question is can we do with photoreceptors? And there's a lot of labs working on this. We do a lot of similar things where we see how are cones born in development, how are rods born in development, and then try to engineer these factors into glia. Because in all honesty, it's going to be a lot easier to make a light sensitive photoreceptor work than it is to make a ganglion cell go back to the brain, right? So there's a tall ask. Okay, so I want to just kind of wrap up the summary here. I hope I showed enough background here to convince you that, with ASCL1, we can stimulate mammalian Mueller glia to regenerate neurons in the mouse retina in vivo. I think it's still a pretty big deal that we're trying to grapple with here, how good this is going to work. And I also hope to show, I think that this is a plastic system, that if we keep thinking about how to engineer these cells to different types of cell fates, we can do that by using developmental lessons. And then, lastly, why these cells are definitely incomplete and they're, definitely from glia and have a little bit of a hangover problem. They're still expressing these exciting characteristics of RGCs at the molecular level, the chromatin level, and the physiological level. So yeah, it leaves us with a lot of huge questions. Are we going to be able to get other types of neurons? Do we need all the kinds of neurons? We know there's 30 plus RGCs. Do we need all those again, or will the brain figure out new outputs? In a different alternative setup? I'm very interested in that question because if you look at how it works in frogs and fish, there might be alternative plans the retina could use to, contribute to useful vision. Do these neurons actually can be used for functional vision recovery? We don't have a handle on that. We're trying to think of ways to do that, at least with the bipolar models, because the ganglion cells, as I mentioned, they don't know where they're going. They send these big axons out. They often go the wrong direction. They don't exactly know where to target. And, lastly, is this going to be a thing that's going to work in humans? And Tom Ray's lab and Juliet Volschlegel have published this year, at least in fetal human and organoid cultures, ASCL1 can make human Mueller glia also neurogenic. So we do think it's going to hopefully scale across the species, but that's also a big question we have left for us. Yeah, and as Michael mentioned, this is kind of my transitional projects and works from my cloudy skies in Seattle to now my snowy skies in Syracuse. But yeah, we had a lot of big help from the Mu Lab for giving us those mice, the PAL4 islet mice that really sped up the project by like 3 years. And, without Fred, we couldn't really show you this physiological data to show these actually make synapses and connective circuitry. So yeah, really grateful to that shout out as well. Yeah, thanks.
Excellent. Excellent presentation. Thank you so much. I mean, I have so many questions here, but in general, I mean, the idea is absolutely fascinating. Of course, to think that we went from thinking that astrocytes and glial cells in general are the dumpster of neurons.
Yeah, exactly.
And now to think that they can actually replace neurons and give rise to neurons and so on. I mean, they're so plastic, so so flexible. That's really very, very interesting. So do you know, you mentioned OTX2 and then you mentioned this combination of islet 1 and POF42, but do you know what kind of effector is downstream to ASCL1?
Yeah, that's a really good question. So I don't have the slides for that, but we do have a paper where we had done chip sequencing of ASCL1 during our generation to see, where does ASCL1 bind to in the genome? So where does it bind to in development, which we're interested in, and also where does it bind to in regeneration? And what we were finding, and that's a whole different story, but it's very interesting. Sorry, I was going to show the slide here. What we were finding is ASCL1 in the glia does bind to these famous sites for development. We know a lot of those. A lot of those are notch effectors. A lot of those are things like OTX2. It binds to the enhancer of OTX2, or the promoter, sorry, of OTX2. But it also now starts to bind to these, what we call inappropriate sites. So ASCL1 does a lot of its famous downstream effectors, but now it starts to bind to places it shouldn't normally bind when we overexpress it. And a lot of that goes into my other arm of my lab, which is a neuroinflammation. And when glia become inflamed, they open up a bunch of chromatin they shouldn't normally have open, that ASCL1 just like stupidly binds to the wrong places. So some of the reasons we think it's not very effective is because it actually doesn't get a bind to its downstream effectors properly. But we do have a huge target list of all the downstream effectors. It's hard to know, which ones are the critical ones to start to follow. I don't know if that really answers your question, though.
No, I mean, yes. I mean, the short answer is that you're still trying to find. I mean, there is no one single category of genes that is activated by ASCL.
Right.
I understand. Another thing that really strike me is this what you discuss about the position of these putative, let's call it putative ganglion cells. I mean, they seem to locate to where more or less the soma of the Mueller glia was. Right?
Yeah.
So it means like, I don't know if this is true, but does it mean that maybe the whole dividing, the cell is dividing and then differentiating into ganglion cells in its location and is unable to migrate? Is that a problem? I mean, what makes these cells migrate into the true location?
Super, super interesting and good thought. So I guess two things. I think I have a picture of that. Yeah, here. So sometimes the cells do make it. Like this cell you can see here has kind of made it through the ONL. And sometimes we will see them kind of make it by the ganglion cell layer. But you're absolutely right. Most of them get trapped in the internuclear layer, for sure. And that's a really good question on a few ways. In development, you probably know this, but the way the cells divide is they divide at the limit membrane out here, right? At the base. They go through their S phase and M phase out here, or the M phase, sorry, out here. So in development, they track this long string of like interconnect nuclear migration. Now, when our Mueller glia divide, we don't know. Does it just divide in the INL and then get trapped because there's so much thick dendrites around there? Like the neuropill is so insanely dense. To me, it's really shocking that a neuron can ever move through that space, because you're going to lack your cues, right? The cues to like do laminar positioning, I'm assuming, is long gone. You had that in embryonic, but why keep the cues of where to position a ganglion cell in the adult mouse, right? So we've been thinking about this a lot, like what are the cues for lamination, and can we re-engineer that as well? We have a paper with Peter Baranoff's lab that was published this year in PNAS, and they're trying to find molecules for that exact reason. Could you in their context, they're trying to transplant ganglion cells in the subretinal space and then drag them through the retina to the RGC layer. And we were thinking, oh, that's perfect. That's what we want to do too. Can we just inject things in the intravitreal space to kind of drag the ganglion cells to the RGC layer? So there's a lot of thought for that. I don't think we have a good handle, but we've thought about some of these chemokines like CXCL12 or something. SDF1 is that paper. But there's also a lot of these efferents and robos and slits, and we're trying to kind of get an understanding of what is the chemotactic cues for lamination. It's not that well worked out, which would be cool to look at. And is that just a big disruption issue that we have to handle?
Right. Another factor in that is also the buildup of the extracellular matrix and
For sure.
cases that break it down and can be activated or not by cells and secreted by cells.
Exactly. And I'd love to work on that. And I don't have a good handle on that. So we show a lot of other data on the other side of my lab, where if we keep the immune cells out of the equation, the neurons regenerate way better. And we think a lot of that is that they lay down this like anti-regeneration ECM. We don't have a good handle on what the molecules are, and that's still kind of like a working hypothesis. But I would love to think about the like ECM of development versus mature retina versus the injured retina, because clearly this is an injured retina, and we think about aging in the future and all of these different things. That's going to be a really big hurdle on where these cells land.
So your model is NMDA toxicity that you inject intravitreally, right?
In the story here, yeah, but I can talk about other models if you want quickly.
Oh, you've seen this in other types of injury.
Yeah, so we know that we can do like optic nerve crushes. We also can do light damage injury, and this will also cause the regeneration of neurons. It doesn't matter the cell fate choice. So ASC1 makes a bipolar, whether you kill the photoreceptors or the ganglion cells, it doesn't matter. The injury stimulus doesn't change the cell fate choice of these cells. And then we have another strategy with no injury at all. Some factors we can just turn on in the glia and they can make neurons without any injury. So that's one of the ways we're hoping to get around this. And then I have a current project now with like aged mice, and we're kind of hoping to do other like RD models, but we haven't yet.
Fascinating. I spoke too much. So is there anyone in the audience that would like to ask something? Any questions there? I think that I muted people. That's my bad. Sorry. Mute all. No, it's okay. So if anyone has a question, please unmute yourself and ask it. Nope. Okay.
Cool.
Okay, last call. Okay. So this was fascinating. Thank you so much for joining us. Thank you, really. We look forward to see more of your work in the coming years. And we invite you to visit us here in Rochester, of course, anytime you like. We do a lot of electrophysiology here. We are not Fred Ricky, but we do some electrophysiology, and if you're interested in that, into recording from your RGCs, and trying to look at that would be awesome. Thank you so much. Thank you for joining us, and have a good day.
Michael. Yeah, thanks, Michael.
Thank you, everybody.